I want to welcome everybody. I want to welcome you all this evening, and thank you for joining us for tonight's Nat Chat. The 2019-2020 season of Nat Chats is made possible by presenting sponsor, the Downing Family Foundation, and media partner KPBS, the public media station serving San Diego and Imperial counties. Speaking of shout outs, I hear that the students of San Diego Workforce Innovation High School are with us here tonight. Good on you, and thank you for joining us tonight. If you're enjoying our Nat Chats for the brief respite that they provide during our current pandemic, then you'll be delighted to know that we got your back. I'm ecstatic to announce that we are getting ready to have some paired talks in Spanish and English on the same subject. So for instance, on August 11th, we're having a panel presentation in Spanish called Las Islas Oceánicas como Laboratorio Global. Speakers will discuss scientific research, conservation, climate change, and biodiversity through the lens of these specific islands off of the Pacific coast of Mexico. It's called the Revihielos Archipelago. And so that's all going to be in Spanish. But then on August 18th, we're going to have an English language talk on the islands called Tales from a Remote Island Expedition. And it's going to be presented by the always engaging Dr. Sula Vanderplank. And she'll be discussing the importance of island biodiversity in addition to walking through the day-by-day life of an island expedition. Now, it's important to note that while these talks are sort of a pairing, they're not a Spanish-English version of the same talk. And so if you're bilingual, or like me, wish that you were bilingual, then tune in for both of them and you're going to get different perspectives and different languages on the same subject. We're going to repeat that style again in late August and early September. And in this case, we're going to hear about a binational multi-agency effort to reintroduce the federally threatened California red-legged frogs to 2 Southern California habitats where they've been absent for many years. Trust me, it's going to be very cool. Same sort of format, one in English, one in Spanish, from different sets of people, same topic, different perspectives, different languages. And if you follow us on Facebook, you can check it out on our online calendar for more details about who the speakers are and what they're going to be talking about and so forth. We're really stoked about it. I hope you are too, and will join us. So if you love this, or you love nature, or you love nature, or you have fond memories of watching the pendulum in the museum knock over little tiny wooden blocks, then boy, do I have a deal for you. You can double a donation right now by giving before August 31st, and then a local philanthropist will match it dollar for dollar, doubling the value of your gift. So make a donation and then pat yourself on the back for actually making twice the difference for us as an organization. We really will truly appreciate it. But Michael, you say, my pocketbook is a little slim these days. How else can I make a difference? Well, if Rob's talk tonight gets you fired up to participate in the Never Home Alone project, then stick around after the talk for an iNaturalist training by the Nats Community Engagement Manager, Lauren Marino Perez. Wonderful human being, and she will train you on how to contribute to some of the museum's projects and the project that Rob is going to regale us about in moments soon.
But to make the important point, you can both give money and listen to Lauren's talk. This is like you
Rob,
give money.
I haven't introduced you yet. So I'm honored to introduce our speaker tonight, Dr. Robert Dunn. I had the pleasure to share the office next door to Rob when we were both working on our PhDs at University of Connecticut. Rob introduced me to many important things over the years, including null models, the wonders of strawberry salsa, and the value of a dollar sign in Microsoft Excel. Since those days in the early 2000s, Rob has gone on to find success in just about everything that he touches. I personally think this is because Rob nurtures creativity in both himself and others. While working on his PhD in ecology, Rob won the Wallace Stevens Prize in Creative Writing at University of Connecticut for 4 years in a row. And that spirit of creativity clearly lives on in Rob's writing. He has 6 books and another one forthcoming in 2021 under his belt, published in 15 different languages, countless of articles in the popular press from National Geographic to Wired magazine, documentaries, awards, scientific publications across a baffling number of domains. His list of funded grants to me reads like something from Roald Dahl's Charlie and the Chocolate Factory. His most current grant title is titled Targeting Superior Yeast Metabolisms from Insects for Improved Food and Flavor Products Using a Proprietary Cultivation Pipeline. And if that's not Wonka-ish, I've not heard of a grant title that is. Truly amazing. Rob's done a lot of things in a lot of places, but mostly while working as a professor, teacher, and beloved colleague at North Carolina State University, where he currently serves as the William Neal Reynolds Distinguished Professor in the Department of Applied Ecology. But most importantly, I think Rob's a wonderful human being. When I was an incoming student to University of Connecticut coming from deep South Alabama, Rob, not knowing me from Adam, replied to an email pleading for help to help my wife, my newborn son, and myself move into a cruddy apartment in the backwoods of Connecticut. It was the first of many kind acts that I witnessed from Rob over the years. He cares about people, he cares about science, he cares about biodiversity. It's my pleasure to turn it over to a caring human being, Dr. Robert Dunn.
Thanks, Michael. I got lost in the end of your introduction trying to think if you're from the deep South, am I now in the shallow South, and how does that all play out? But thank you so much, Michael. It's a real honor. I've had the great chance to visit your museum, and I'm very earnest that people should donate, be engaged in any way you can. I'm shivering a little bit now because it's so hot outside here that the university decided to turn the air conditioning up way high, so I apologize for shivering in advance. I'm going to talk to you today about the life in homes, but where this really starts is back when I was a graduate student with Michael at the University of Connecticut, and we were trained to do very basic biology. And by basic, I mean we were supposed to go out into the world and figure out its pieces and understand the rules that governed how all those pieces came together. And so it was a wonderful, wonderful place to be a student. It was a place where people just loved the natural world and understanding it. One of the other people that was in our little corner of the building is this guy, Peter Neskrensky. This book, Relics, is one of Peter's books. And so we spent all this time just getting to know nature and figure out nature and to figure it out for its own sake. And so that's what I was trained to do. But as I went through my career, I ended up in a place at North Carolina State University where everybody in my building can do something useful. They can make more fish, they can make more wheat. And for most of my career, what I could do was to tell you about obscure insects living in faraway places. And I love that, but I would go and give talks. I would talk about ants, and people would say at the end of my talk, you know, that's nice, but what do I do about the ants in my kitchen? And I hated that question because I study ants, I study insects, I study microbes because I love them. I think they're beautiful and fascinating. And the last thing I want to know about is how to kill the ones in your kitchen. But at some point in my career, I started to think about, well, what do I know about some of these species? And what sort of foray could I take, if not into your kitchen, sort of moving in that direction? And one of the groups of species I was pretty intrigued by, in part because of Peter, was orthopterans. So crickets, katydids, and their relatives. And one of those orthopterans is a thing called a cave cricket or a camel cricket. And there are lots of species of camel crickets. I knew about them because of Peter. He found one in my basement when he came to visit. And so I thought, well, what if we do something where we just survey people to figure out, do they have these camel crickets in their basement? And we survey people across North America, and we'd figure out, like, where are these things? And it was kind of this sort of attempt to tip a little bit of what we were doing toward houses, toward daily lives. And this first image that you're hopefully seeing now is actually a cave painting on a bison bone of a camel cricket. And I love this because it's an indication that people have been thinking about these camel crickets for a long time. They've been living with them for a long time. And so I thought it'd be fun just to figure out, well, where do they live? And so this is the thing that I thought we would see. It's a native camel cricket species. There are a number of relatives of this species that are either live in caves or maybe make their ways into houses or live outside in the leaf litter. And so what we did was to ask people, we showed people a picture, do you have these? And this is work led by MJ Epps, who's a very, very fine scientist and by most accounts a finer fiddler. And MJ sent out this survey, and almost immediately we got results back. And what you can see here are some of the results. So the white dots are people who said they didn't have camel crickets, and the red dots are people who said they did. Now, this was super interesting, but it was wrong. Because what we know as scientists is that in 1965, there was a great big thick book written about camel crickets that told us exactly where they should be, and this is not where they should be. And so the first response of scientists when something is wrong like this, engaging the public, is to think the public did something wrong. And so people thought, well, maybe people are looking at cockroaches in their houses and they think they're camel crickets. Maybe people in California, even if they had a crawl space, they were scared to go into it. And that something like that happened. And so MJ would then ask people for pictures. Send us a picture of camel crickets in your house so we know what you're looking at. Let's make sure we're looking at the same thing. And when people did that, we were in for a surprise. And the surprise was that instead of the native camel crickets that we expected to find, we instead had pictures sent to us of this giant Japanese species. Now, this one's not quite to scale. This is a picture taken by Peter many years ago, but they're about the size of my thumb. This is an ordinary human-sized thumb. It's not quite as large as Michael's. So it's in between my thumb and Michael's thumb. Can you hold yours up again, Michael? Can I see you? Yeah, right. So it's between those sizes. So it's a big animal. And what was amazing was that we didn't see one picture of this camel cricket. Basically, the vast majority of the camel crickets people were reporting were this introduced species. And so the reason that they were in the wrong place is that it was the wrong cricket. And so what had happened, it's not, there's no judgment there, but this camel cricket had moved from tropical Asia or temperate Asia, tropical Asia at the boundary, moved house to house across North America, and basically that had happened unnoticed. And this to me was like a big wake-up call that if we're not noticing a thumb-sized camel cricket in our basements, in houses in North America, what else might we be missing? And the second thing, let's see. The second thing that we revealed while doing this, or the MJ really revealed, was that there was actually a second species of Japanese camel cricket that had moved into New England. So if you see those white dots in New England, that's another species of camel cricket that was in houses and we didn't know was there. And so this was super exciting to me. This was like, whoa, like what else can be in houses? What should we do? But I'm actually not that good of an entomologist, and at least not in the sense of like just going into people's houses. And Michael's laughing because he knows, and figuring out what's there. And so I needed some help to think about what to do next. And so teamed up with my friend, Dr. Michelle Trotwine, who's at the Cal Academy, not so far from y'all. And so Michelle and I, we were having a glass of wine, talking about projects, and decided maybe we could just go into a bunch of houses and survey everything and figure out how many arthropod species are in houses, what's being missed, what's there. If the camel cricket is the tip of the lifeberg, what's the rest of the lifeberg? But Michelle is a great fly scientist, but she also is not that good of an entomologist who can ID everything. And so we needed Matt Bertone. Matt Bertone can identify anything. And so that together we would embark on this endeavor to just go into a bunch of houses and find all of the species we could. And then, as is my wont, I promptly disappeared after we started this project to Europe for a while. And so the project actually started with me gone. And so we didn't know what would happen. And my house was then volunteered to be the first house. And so we would then survey, where we is royal because I wasn't there, what was in my house and each of a bunch of other houses. And this started as kind of a small project. And then something happened that made me think it might be more interesting, which is that I went to my colleagues one building over in the entomology building and asked what we would see. And they all said that we wouldn't see very much and it would be boring. And that to me is a good clue that you're onto something, that if a large group of people agrees you will see nothing, you're probably going to see something. And so what people thought we would find is bed bugs and houseflies and German roaches. And it would be boring. And in fact, one of my dear colleagues actually stood up in a talk and said, this will be boring. And so we went and we tried to figure out, what would we find? Would it be boring? And I'll say I didn't quite know what we would find. I thought, for example, with spiders that we might find like I knew in my house we had this long, skinny Don Quixote kind of spider and this fat Sancho Panza spider. And so I thought, well, I got at least 2 spiders. I don't know what else is going to be there. And I thought maybe we'd find 4 or 5 things per house. And I get an email from Michelle, and she says, good news, we finished your house, and we can tell you how many species there are. And I said, well, what's the answer? And she sent me an Excel spreadsheet with dollar signs, Matthew. Michael, I'm looking at the wrong thing. And in it were 100 species of arthropods. And so my first thought was that Holly Menninger, who was living in my house at the time and works at another museum, that she had just been super messy while I was gone. But then the other houses started coming in, and it turned out that the average house in Raleigh, North Carolina had about 100 species of arthropods and about 8 to 10 species of spiders, so many kinds of flies you couldn't even, we still not ID'd most of them. And that collectively across the first 40 houses, we found more than 1,000 species and probably more like 2,000 species. And it was all kinds of cool stuff. So sure, there were mosquitoes, sure, there were some house flies and some green bottle flies and some house centipedes, but then just many, many more species, even of common things than we would have thought. And so we had, 10 species of ants in Raleigh, and the things that everybody thought we would see lots of were actually very rare. And so between this and the camel cricket, I thought we're going to publish the papers on this and everybody's going to be so excited because after all of my time in the tropics, finally now I've told everybody something about their kitchen. And so now I can answer that question, what about the ants in my kitchen? And the answer is that there are like 10 species and we're starting to study them. And so we published this paper, and that wasn't really the response. Instead, the response was, what good are they? And so my heart just broke just a little bit as I thought about this. And so my first sort of response to this was to think about, well, what do they do? And it's not what good are they, but it's sort of what is a role that they have. And so we looked at this in part. And so a lot of these species clean up things in your house. And so you've got book lice and ants and where are we? There's a collembolin up the top. It's a springtail. Do you see that? So on its backside, it has a little pogo stick. And so these hop through your house and they're teeny tiny. You don't see them and they're cleaning up things for you. And so these are kind of like kitchen undertakers. And then you've got all these kinds of spiders and centipedes. And so you have these functions that are being carried out in the house. But that's not what anybody meant with regard to what good are they. They wanted something more direct. What are they doing for me is really what that meant. And so again, breaks my heart a little, but I started thinking about this. And one of my biases as a basic biologist is that I learned that in thinking about nature, there are no goods, no bads, just truths. That you go out into nature and nature is, and it's our job to understand it. but it's not our job to make it ours. It's not our job to relate it back to our own daily lives. But then I thought again about the people in the office next to me making more fish. And I thought, well, maybe I can do a little better on this. And so my first thought was, well, spiders are eating other species in your house. Many of you said that the species you would be most excited to see in your own house were species of spiders. This happens to be a social spider. There was actually a project in South Africa for a while and a matching project in Mexico to try to figure out, could you bring social spider webs into children's nurseries to control flies and prevent contagion of those kids with nasty things that flies carry. And turns out it works super well. There was even a project to bring social spiders to France to spread them around France. It didn't work as well because they forgot to feed them for a while. But this is one thing that the arthropods in your home do. The predators control other species, and that includes the spiders. It also includes this thing. This is one of my favorite. This is a parasitoid that lays its eggs inside the living babies of cockroaches in their egg cases. And so the babies of this wasp then hatch and eat the living baby roaches. And if you don't like roaches, this is like the key part in the hero movie that's happening under your bed. And so you've got to love these, and they're super common. We find it in the vast majority of houses. And so you might not have roaches, but you very likely have parasitic wasps that lay their eggs in roach egg cases, which might mean you have roaches. But then the other thing we started thinking about is like these are services provided by these species directly to your house. And so if you have these wasps in your house, they're helping you out in your house. But what about broader services to society? And I happen to be a big university where there are lots of people who do useful things. And so I started to think about, what use could some of these species have in a broader context? And so I started thinking about those camel crickets. What good could they be? And well, the camel crickets we know are really kind of cave-like animals, that they're eating really hard to break down foods that are super hard to digest, foods like those that other cave animals like these that you see pictured eat. And so we started to wonder, could the guts of those camel crickets have special microbiome, special bacteria that maybe are super adapted for breaking down hard to break down things? And so that was the end of my idea. And so I needed help. And so I went to the building next door, and that's where Dr. Amy Grundon, my colleague and friend, works. And Amy is an expert at finding microbes to break down hard to break down stuff. And she'd been working on this stuff, black liquor, which is the waste product of the paper pulp industry. And so if you're using paper, you're also making this stuff. And so this stuff in the US is typically burned, or if nobody's looking, it's poured into rivers. And so it would be great to be able to break it down. And so Amy said, why don't we try and see if we can find any microbes in camel crickets' guts that can break this down? And so we had a student, Stephanie Matthews, who started to work on this. What Amy didn't tell me is that this stuff is really lignin, the hardest stuff of wood. Like my table, you can't see it here, but it's wood. What makes this hard is lignin. Lignin is super hard to break down, and for the most part, only fungi break it down. And so the odds that we'd find something in the camel cricket guts that would break this down were super low. And this is in an alkaline bath. And so it's sort of both hard to break down and toxic. But we thought we would try, and I was trying partially because I was ignorant, but sometimes that gets you a little bit of a way. And so Stephanie started a project to see if she could find something. We took one camel cricket from my house. We extracted microbes from that one camel cricket. Only one camel cricket was harmed in this project, and it might have already been dead. I don't remember. And from that one camel cricket, Stephanie doubled the number of bacteria species on Earth known to be able to break down lignin. And so super cool. This camel cricket, which we didn't know was in North America, has microbes that can break down the waste product in the paper and pulp industry. And so this got me thinking, well, geez, I work at a campus full of people who do useful things. What other things could be going on? And about this same time, and the order may be a little wonky here, I got an email that said, Rob, we want to make a new beer for a science festival. Do you have any interesting microbes that we could use teaming up with a brewer to make a new beer? And at about that time, Ann Madden had joined our lab, and Ann worked on wasps, and she worked on microbes and social wasp nests. And Ann said, we've got to try a wasp nest yeast to see if we can make a beer. And so we got some yeast that we found from a social wasp and a yeast that we found from a bumblebee. And the first yeast from the social wasp actually came from Ann's porch, I think. And there was some fiddling to get all this stuff to work. But relatively quickly, the first sort of official try of this yielded a yeast strain that produced beer. And not only that, it could make a sour beer in like 30 days rather than years. And so my part on this was the thinking about who to put together. The hard work was John Shepard, who you see here, who did all the brewing, and Ann, who thought about where to look. But it made us think, well, what else can we be doing? And here Ann is with the keg of that beer. It was kicked quickly. And you can now buy a beer made from this yeast in some stores in the US. So we started doing more and more of this kind of work. Another example, we started thinking about, well, is there anything useful like in your refrigerator, in your basement? And this just led us down all these paths, many of which were fruitful. One of which was to think about, is there anything on fish that we could use to do something useful? You've got old fish in your refrigerator, what can you do? It led to a partnership with Ben Reading, who just happens to be across the hall from me, and Ben's one of the people who makes more fish. And so we started to look at caviar, and could we find new microbes that would make a better caviar? And lo and behold, we found some on fish skin. That's not Otzi, this is Ben, but this is to remind me, but they kind of look similar. That's Otzi Ben. That the microbe that we found that works well to help preserve caviar and give it better flavors also happens to be the microbe that was most common in the body of Otzi the Iceman when he was found. And so he was preserved with the same microbe we're now trying to use to make new caviars. Another example, we thought we could learn from the exoskeletons of household ants to make new textiles. This is work led by Clint Pennick. It didn't quite, so imagine like gowns that were, had amazing texture that was antimicrobial or pro-microbial. That didn't work. But Clint, working with some students in design and textiles, and Adrian Smith realized there were these amazing patterns on ants, and you could use them in a design context. And so they made these new pillows that you can now buy based on the exoskeletons of ants you can find in your house. Omar Halawani looked at another group of ants that are common in houses to find if they're producing antimicrobials that might be useful in controlling antibiotic-resistant bacteria. And Omar's found a couple of interesting features of some of these ants that seem to be antimicrobial in a useful way. Megan Temes works on face mites. She's now in San Diego. And I love face mites. Michael and I got to know them at the University of Connecticut, where Clifford Desch did a lot of the foundational work on these mites. They live on your head. They come out and they mate in your face. They live on your back and your butt too, but nobody wants to hear about butt mites, and so we call them face mites. And they're a classic example of like, who cares? What good are they? Maybe they're useful to your skin. We don't know. But one of the insights Megan had was that on most people, these number in the tens of thousands, and we don't generate immune responses to them. And so it seems very likely they're producing immunosuppressants. If that's true, we need new immunosuppressants. Could they be in these mites? And we've tried to find a good partnership to look for those immunosuppressants. We're not there yet. But that's the kind of thing we can do. And there are more and more examples of this. And so to me, all these things are interesting just because I find them interesting. How could we not know more about these species that live in our houses? If we don't know about them, we really don't know about anything in the living world because these are the species we see most often. But I think it's also useful to realize that it's only once we know they're there, only once we start to study them, only once we ask questions about what they could be used for, that we can start to rely on them. And this extends to smaller things. In salt shakers, we find extremophile microbes that previously were only known in salt flats and hot springs that could be useful for a whole series of industrial uses. Some of those may be thousands or even millions of years old. They actually live inside the individual salt crystals. In drinking fountains, Carlos Galler has now found microbes that precipitate gold. I've heard that could be useful. And so we're doing all this work, and as we do it, I've always been very interested in microbes. We start to wonder what's the even smaller stuff we're missing. So if we're missing camel crickets, what are the smaller things? And so we started to team up with Noah Fear at the University of Colorado to start to look at what's in dust. What are these more minute species we might find in a moat of dust? We did a big project where we worked with people to sample all of these houses you see here. Each dot is a house. And people would go and they would swab their door frames and other habitats in their house. They would send us the swabs. And we would do something like Leeuwenhoek, the first microbiologist, did, but instead of looking through a microscope, we would break open the individual cells of microbes from the dust. We'd make a bunch of copies of their genes, and then we would match those genes to name species. And so we could do that for bacteria, but also for archaeans, which are this other single-cell life form, for fungi, for plants, for animals. And we could do sort of CSI, kitchen, bath, and beyond. We started with 40 houses. We eventually got up to the 1,000 houses, really thinking about the house as a full habitat and sampling these different parts of the house. When we began to do this, we found thousands of species of bacteria, many of them not named, many of them new to science, all but a handful of them never studied in any detail. Just for a little context, these are archaeans. So archaeans, a single-celled life form, as distant from us as our bacteria, as ancient as our bacteria. When I was a grad student with Michael, when we both didn't have any gray hair, when I had hair, I used to have really long hair when Michael and I met. But now it's all on the back, but that's separate. But way back then, the number of archaean species on Earth is this small bar. And now the number of archaean species we've found in homes is this big bar. When we looked at fungi, this is the number of named fungal species in North America, so like 25,000 species, give or take. This is the number of fungal species we found indoors. This isn't because there are so many species indoors, it's because fungi are so unknown and we're using a new method to see them. And so what this is an indication of that we're surrounded by fungi we've not yet named, much less studied in any way. Right now, you're breathing in hundreds of unnamed fungus species. When we looked at bacteria overall, I like when Michael claps. It makes this all worthwhile. This is the number of mammal species, number of bird species on Earth, number of bacteria species we found indoors. So really huge numbers. It is worth pointing out in the time of COVID, which is a virus, not a bacteria species, that fewer than 20 of these species that we found are potential pathogens. And so you're surrounded by all these species, and just a teeny handful are problematic. And so it's akin to going out into a forest and there are dangerous species in the forest. In the same way, just a teeny handful are really problematic. And it's just like in your house. We can then do cool things in houses, like we can make a kind of map, which is what you see here, where the closer two sample points are together, the more similar they are. And so nobody likes this one except everybody likes it. The blue shows door trim. And so the door trim sites tend to get outdoor microbes coming in. The greenish things are kitchen associated sites. And so what you see here is there's like one TV set somewhere that's got like kitchen-associated microbes. This is like somebody who threw a hot dog at the TV during a football game and it stuck. I don't know, something of that nature. And then the top right is what nobody likes at all, is that pillowcases and toilet seats relatively indistinguishable. Just is, we're falling apart, and we fall apart where we sit, and we sit different places. And so we can also then do things like figure out what determines what's in your house versus my house. And we've done many versions of this kind of study, and overwhelmingly the biggest, most important explanatory variable is do you live with a dog? In houses with dogs, there are tons of these specific microbes that are much more common, and they're common even on things the dogs don't touch, like your TV screen. So the dog is just dragging these microbes in like a big dust cloud, and they're going all through your house. In people's houses who recently have a dog but don't anymore, we still see the effect of a dog. And so it's a big effect. And so overall, we see an effect of pets, women versus men. Men's houses are full of armpit microbes. The food you eat, if you smoke marijuana, we see plant DNA from the marijuana floating around. Ventilation, like do you open your windows? Do you have an AC unit? But any story I could tell you about this is going to be partial, because the truth is what we need to do is natural history. We need to do basic biology again in our houses. And each of the tens of thousands of species that we study is going to have a different story. And, that's a problem. From a medical point of view, it's a problem from a public health point of view. But from a discovery point of view, it's that if you're a 15-year-old interested in science, that most of what is knowable is not even discovered yet in your house. But what's the big picture? How have our daily microbes and arthropods changed? Is your house normal? Are our houses like what they would have been thousands of years ago? We've just started to study this, but we're seeing elements of the story, and we know some of them already. And so, one of them is that as humans moved into big settlements, we got a bunch more sort of bodily microbes that turned up with us that caused problems. Another change that happened is with germ theory, we started to realize some small species could be bad. And so we started to view things that were small as negative. This is a great image I love from the sort of early moments in understanding cholera. This is the Thames, and it's what's in its water. And so there's a point when we start to see all of it as bad. And in response to that, we do a bunch of sensible things. So we figure out how to make drinking water not free of microbes, but free of pathogens. So clean drinking water saves millions of lives every year around the world, and the lack of clean drinking water still kills. Vaccination, washing your hands with soap and water. All of these things had huge beneficial impacts and are really simple interventions. But at the same time, we also did all of these other things that potentially change what we're exposed to. We built giant buildings. We sealed those buildings. We built buildings that the windows don't open on. And so what's the net effect of those changes? And so we don't have a super clear picture of this, but I'm going to give you two quick vignettes about what we do know. And one vignette relates to what lives in chimpanzee nests, which we might imagine is kind of what used to live in our own nests, say 6 million years ago. And the other is what lives in space stations, which I think if we're imagining, the space station now, if we're imagining a future for ourselves, we often think about something like the space station. If I just clean my house enough, if I seal it off. And so what do those two things look like? Well, the space station, we now know thanks to a bunch of sampling, the International Space Station, it looks most like a toilet seat. So based on its bacteria, it looks most like a toilet seat. Based on its fungi, it looks like a dishwasher. And so on the place we've most cleaned, we've most sealed things off, we've most tried to prevent contamination. It's still full of microbes, but it's full exclusively of human microbes and things that live on plastics. And so if you imagine that you're going to clean your house fully and get everything out, this is what you're going to make. You're going to make a scenario that looks just like a human body falling apart. At the other extreme, what a chimpanzee nest look like. And I imagine when we started this project, this was led by Megan Temis, who I mentioned earlier, that it was going to be full of chimp feces and chimp fecal microbes. I mean, just look at that chimp right there. It's not wearing pants. What's happening? Something awful. And so we thought, well, what are we going to find there? And what Megan found working in the Isa Valley in Tanzania, it was that the chimpanzee nests there were dominated exclusively by microbes associated with leaves, soil, roots, and bark. We saw basically no trace of the chimps, the chimp microbes, or the arthropods associated with chimps. And so if you had to characterize the shift that's happened to us, we've gone from daily lives enmeshed in nature to daily lives enmeshed in our own falling apart. And I don't have a ton of time to talk about this today. Some of it's in the book, but it's also elsewhere online if you want the free version. But there's an emerging consensus that having done this, that we lost something. That more diversity in some way was good. And that when we have less diversity, we're losing beneficial species that help our immune systems work, that help our bodies work. And as a result, we're seeing in kids that don't grow up exposed to nature, exposed to a diversity of microbes and other species, a whole suite of autoimmune and allergic and disorders that are becoming much, much more common. Super, super complex, but it seems to be associated with this shift. And I can come back to this later, but there's more and more evidence. And so what's the answer? What do I do about the ants in my kitchen? And I think part of the answer is super dissatisfied, which is that we need moderation. People hate moderation. Moderation is like the hardest thing in the world. Like, what does it look like? But we need to try everything we can to kill off things we know are pathogens. So wash your hands with soap and water, wear a mask, get vaccinated, do these things that we know help. But at the same time, when you don't need to kill the species around you, don't. And so there's this target the things you know can harm you. And washing your hands with soap and water is a great example, because what that does is it gets the stuff that's just landed on your hands and removes it, but it doesn't change in any way the microbes actually on your hands, your good microbes on your skin. They remain. And so we need moderation, and I think we need wonder. I think wonder goes a long way. But I'm going to quickly say, and I realize I'm getting a little bit close to the end here, so I'll be quick about this, that when I get to this point in talk, often there's still this sentiment of like, well, I don't have kids or allergies. Can I just kill it all? And I think the first answer is the space station, which you just can't. It's just life is more powerful and wonderful and ever present than you are a control freak. But let's imagine you still want to try. When we do this hand sanitizer is a perfect example of what we do. It kills 99.99% of germs. I am here to tell you that 0.01%, those are the real jerks. And so if you kill 99.9% of the germs in your house, this thing, the advertisement tells you what you're left with. You're left with 0.01. And it's non-random who you're left with. And so this is what happens with insects, right? So if you use pesticides all the time in your house, they kill the species that are susceptible, and what's left? Well, what's left are who's resistant. And so the pesticide chlorine was first used in homes in 1948. By 1956, some German cockroaches were resistant. By 1966, both houseflies and German cockroaches were resistant to virtually every pesticide, including DDT. And so they're the 0.01%, and they are loving it. And what's even more amazing is we thought we got super clever and invented roach baits. And roach baits, they have a little sugar, and so you have to go in and you get the sweet, and then you get the bait. And so it's much harder to evolve resistance for a variety of reasons. And that was working great until it turned out that some of the German cockroaches seemed to just not like roach baits anymore. They were like, it's not for me. And so Ayako Wada Katsuma here at NC State did amazing work in which she was able to show with Jules Silverman that what had happened is that those roaches had evolved resistance to the baits, and the way they'd done it is that natural selection had rewired their antennae so that they perceived basically the sugar in the baits as bitter. And so they would go to the roach bait, they would go, "Oh, is it sweet? No, this is terrible," and they would walk away. And so in terms of evolution, this is what you're up against if you want to kill everything. But to me, the even more powerful version of this is an experiment done by Michael Boehm at Harvard, and this is where I'll finish, and I'll quickly turn to Brett and then pitch over to Michael. Michael made this giant petri dish, and on either end, he introduced bacteria, E. coli, that were not resistant to antibiotic. They're super susceptible. These are like the, good news, and then we can kill them. And then he made a set of columns with increasing concentrations of antibiotics to figure out how fast, with no genes for antibiotic resistance, could they evolve resistance. And so this is like you take too many antibiotics, what happens? And so I'm going to show you, not in real time, but sped up a little bit, what happened over 11 days. And so here's the giant dish again. In the middle is this super high concentration of antibiotics. And so this is the video of what happens, sped up. And so first the bacteria grow from the end and they get to that first level of antibiotics. And that's kind of like a wall. And they stop for a second. And then what you're going to see is, look, there's the first individual lineage of bacteria, a mutant that makes it through with resistance to the first concentration. Oh, look, there's so many more of them. Just incredibly fast. Now they get to that second level of antibiotics, and a subset of those lineages makes it through. But remember that middle is the fortress. This is antibiotics. We're just going to hold back nature by killing all of it. What is going to happen? Boom. There they go. In 11 days, again and again and again and again, this happens. Which is to say, if we just try to kill everything, it doesn't work. It doesn't work for a whole series of reasons. And so we need some way to have moderation. And so if not this future where we just kill everything, then what? Moderation's hard. I've actually forgotten why I have this giant baby in my talk. I'm sort of alarmed by it. As I was trying to figure out where we might see a kind of solution, I started talking more and more to bakers and getting interested in what bakers were doing with microbes in their homes. And one of the things bakers do is make sourdough bread. And sourdough bread's super simple. You put flour and water together, and yeast and bacteria colonize it, and they start to bubble. The yeast produce CO2, the bacteria produce acid, and it rises, and you can use it to leaven your bread. This is what all breads used to be like. And then we industrialized the production of one yeast strain. I know I'm late. I've just got one more slide. And so this was the thing. This, in essence, is what we want, because what happens here is when these microbes produce acid and CO2, they kill other microbes. And so this is a garden that weeds itself. If your sourdough is acidic, it's safe. And so this is a model for what we might think about in our houses. How do we make a garden that helps us keep pathogens at bay rather than one in which we're trying to kill everything? And so I'll pause there on this model, and we can come back to it a little bit in question and answer. And one of the interesting things is that we can come back to a little bit is how doing something like this actually changes not only the microbes that are in your house, but on your body and even all around you. And so the act of moderation, the act of favoring some over others, changes what's in your food, but also can change you. And so I'll close there. Apologies for going a couple minutes long. Is anybody still out there? I don't know.
I'm here, Rob.
Oh, hey, Mike.
I got you. That was fantastic, man. Totally incredible. You had me in stitches. I learned a lot. I'm definitely a big believer in the middle path. And so I love the message of moderation. I have to start with a question that we got over the Q&A here that is someone after my own heart. Heather Sabin asks, "What's that beer called?"
I can't say, but you can look. Well, no, it's a bummer to me too. So you can look at things happening from the company Lachancia LLC.
Okay, we'll look them up. We need that wasp beer here in San Diego.
Yeah, no, I agree.
So there's a couple of people have chimed in and like this question from Nelia Sakurty. Is it good to be a bit dirty to increase microbe exposure? Is washing too much negative? And hold up, the question disappeared on me. And is it more important with children growing up, and as adult, does it matter quite as much?
Yeah, those are super good questions. So one version of the answer is stuff that we know is good. So like if you live someplace where your air outside your house is not toxic, like opening your windows more, gardening more, getting in the forest more. These are all things we know increase exposure to good soil and leaf microbes that aren't dirty in the way that we think of like a hygiene dirty, but we see benefits from them. And there are now good studies of a variety of kinds showing those benefits. And in the same way, getting dirty in fermented foods is another thing. You do still want to wash your hands with soap and water. It works. It's so super effective. But there's now a fair amount of questioning, like how often should you be taking a shower with a bunch of products, and what is that doing? I think it's an open question in some ways. I think probably we'll find in a number of years that the frequency with which we like super scrub bathe is disfavoring some microbes that we need. But we've also created cultures in which like that's the norm. And deodorant antiperspirant is another thing like that. Like antiperspirant actually favors Staphylococcus bacteria and disfavors the slow-growing, stinky microbes that gorillas and chimps use for signaling. Mm-hmm. But we've created a culture in which armpit stink is viewed pretty negatively, and so if you're the only person making these kinds of changes, there's a social context that I think is real.
Sure. Yeah, and that's part of this moderation thing, this idea of moderation, right? Is that,
if you're just at home with Allison, you can stink all you want, Michael.
Yeah, totally. Absolutely.
The other piece of that question, though, is about young kids. And so I think it is true. It matters less for people as they get older. But but maybe the one way to think about that is why might that be true? And so one reason it's true is that your immune system is kind of making sense of the world. It's like it's learning when you're really young. And so that's one part of it. Super complex. Anybody that tells you we understand it well should not be trusted to sell you a car. But that's more when you're younger, but it's not done when you're younger. Your immune system continues to learn the world.
Right.
The other part would be that you acquire your good microbes when you're young. And so a lot of that happens during birth. So you get vaginal microbes, you get fecal microbes, you get skin microbes from your mom if the birth is vaginal. If the birth is C-section, most of those microbes are coming from chance encounters with the mother or from the hospital. And so in either case, the window for acquiring those microbes and the skin, we don't know how long it is. In the gut, it's really framed by when the stomach becomes acidic. And so in babies, the stomach is not acidic. And so things that the baby ingests very easily become established in the gut, in the intestines. Wow. As you get older, that window closes. That appears to be very human. Our stomachs are like turkey vulture stomachs. We have very, very acidic stomachs. It looks like that's different for other primates.
Huh.
And so that's the other piece. And so the tricky part, so I would say, like, in the context of being public health dirty, in the most cases, you don't want to be, right? But if your baby is born C-section and doesn't get the right gut microbes, there are only so many places they can come from. Mm-hmm. And so that's actually a tricky thing that the medical community is going to have to come to terms with. Right now, those microbes are being acquired by chance from fecal microbes in the environment, fecal microbes from mom and dad, and fecal microbes from dogs, with no control. What would it look like to do something better there? And we're not even having those conversations because I can't see the whole audience, but I can tell people are giving me a look right now. It's a hard conversation, and yet it's super important. And then and I'm going on, but the other time it's really important is as people get older. And the reason it's important then is that your stomach acidity is reduced again. And so if you're over 70, your stomach is getting closer and closer to neutral. And so at that point, you actually become very susceptible to foodborne pathogens because your stomach is no longer acting as much of a filter. And so these different time periods, the balance is different.
Yeah. So Tom Smith has a question that I think you possibly just answered, which is, do you remember why you had the picture of the giant baby? Was it related to all this that we just discussed?
Yeah, it was.
There we go. Asked and answered.
Well, it's also to think about like how colonization, and it sets up with the conversation I didn't get to with the sourdough bread, which is we did an experiment with bakers where we were trying to figure out, do the bakers' hand microbes contribute to their starters? And they do.
Oh, that's
but
so
the
cool.
other thing we found is that the bakers have hands that are covered in sourdough microbes, even after they shower. So the act of being a baker changes their microbiomes.
Oh, wow.
And to me, that's really, it's really a challenge, and it's really special. And it's a challenge in the sense that to know that every day the way you live your life is changing your microbes. It's defining who you are in some way. What do you want it to say about you?
I love it. So, I hate to turn things to intercity rivalries, but there is a little bit of an intercity rivalry between San Diego and LA. And from the perspective of your talk tonight, can you talk a little bit about the big difference? Is there any difference between San Diego and LA?
So we don't know yet, but my expectation is that we find way more interesting arthropod species in homes in San Diego. Yeah, I think, LA, it has its charm, but it's quaint village that it is. But I think, so this is a climate and biome map of North America. And if you look down at San Diego, you see like you're really at the intersection of these different biomes. You have this extreme desert, you have these other ecosystems. And so the potential for really different species to be present there. But the other thing it is kind of it's kind of a climate gateway. And so if we're seeing new species moving into North America, species we didn't know were here, San Diego is probably the best city in North America to study.
Yeah.
And so earnestly, I mean, I would love to have more data from San Diego, from the arthropods in people's houses. And my guess is we're going to see new species, new to North America, maybe just new.
Yeah.
And we have some data, but not tons. And so I think it's a real opportunity. And I'll just take this as a chance to say too that Michael and I are working on with a bunch of other people a new version of the project we did in homes, where you can now take pictures of the arthropods in your home via this Never Home Alone platform on iNaturalist, and they contribute data points to the overall project. and allow us to see what's in homes around the world. And what we're seeing already is that, we started in Raleigh, but different places are super different. And each place seems to have different new species we didn't know were there. And this is an old slide. You can tell because now we're at almost 15,000 records in houses. But it would be really great to get in this project more samples from San Diego. And I'll just quickly say that Lauren Marino Perez is going to talk later about how to do this. I'm supposed to say this at the end of our conversation. Michael, I ruined it. Sorry, but
no, it's all right.
I'll show it again.
There's an interesting question here that is very timely and relevant, and from Luca, who asks, what are your thoughts on how to best balance COVID-related hand sanitizer use with responsible microbe conservation? And I first just want to point out that I love it, the fact that Luca just came up with this phrase of responsible microbe conservation. But how would you answer that question?
Yeah, that's a pleasing question, Luca. It's a pleasing statement, or I don't know, it's a sentiment. So it's tricky, right? That the part of moderation is knowing that there are both species we want to actively control and times we want to do more to control them. And so, especially if you have to go places where you're meeting other people, where you're touching other doorknobs, that washing your hands with soap and water is super important right now. And if you can use soap and water, it's better. It does a better job of keeping your microbes intact and still getting the virus off. But then there are some cases where you're going to use hand sanitizer. Don't spray your whole body with hand sanitizer. I would discourage it. But your hands when you need to. The other thing I would say is that the alcohol-based sanitizers work fine. The ones with triclosan don't work any better than any of the other ones and have a much higher risk of triggering some resistance. And so if you can use soap and water, if you can't use alcohol-based sanitizers, and then if you're given no choice, right now it's still better to use something because COVID's a beast.
Yeah.
And yeah.
Yeah. Thanks. Ashley Salas asked a question that I'm sure you must get a lot is, how do you deal with your own home? Do you permit insects to be in your home to stay, or do you remove them? And she didn't ask this part, but I'll extrapolate a little bit, like, when and do you or don't you use pesticides, et cetera?
Yeah, so I mean, mostly it's marital, mostly the answer to this question, which is that we keep species in the house that my wife is fine with, but she doesn't like house centipedes.
Yeah, they're creepy.
And so I tend to take them to my office and let them run around. But I try to make sure the house is sealed so we're not getting extra stuff. If something's coming in that I know also feeds on garbage, I try to control it using sort of, non-pesticide based measures.
Sure. Yeah, exact same thing in our house. It's like, there's plenty of things that that come into our homes that don't need to be there necessarily in the density that they are, and just use common sense, moderation.
Yeah. Yeah.
Go ahead.
That's it.
Someone asked, and I'm having trouble finding it back here. Okay, here we go. It's Jose Mendoza has asked about species in pantry, car versus shoes. And the reason why I bring this question up, I know you can't answer maybe the specifics on the number of species in our pantries, but I like this idea about cars. Have you guys looked at like what life is like in cars?
We maybe I'll do two parts. One for pantries is we know a lot, and what's really cool about pantries is they're disproportionately dominated by species that moved into homes no later than 2600 BCE in Egypt. And so I have a colleague, Eva Panagiotopoulos, who's at the University of Edinburgh, and so she works in archaeological sites in Egypt, and she works on insects. And she's found most of the species that are common in pantries already present there.
Wow.
And so, pantries are really interesting in that way. And then we created this stored food environment and species actually evolved to take advantage of it. And so Psittophilis grain beetles, which are common in pantries, they lost their wings because they didn't need them anymore. And they acquired special microbes because our food's not very nutritious. and so they use those microbes to get vitamins they need in response to our starting to store food. And so pantries are amazing. And we've just started to study them. But if somebody wanted to study something in detail, beautiful studies. Cars we've not looked at. My guess is it's mostly really small stuff, so mites. But there are tons of kinds of mites. And, dust mites are common and relatively diverse, but there are also these things I call leopard mites that are predatory on dust mites. I bet they're common in cars.
Uh-huh.
Yeah.
Okay, Rob. Well, this has been truly fantastic. It was a pleasure to see you again, though it was from a more than socially distanced way. So in closing, can you give the folks at home some ideas about ways that they can participate in the various projects? I know you talked a little bit about the Never Home Alone, but I know your lab's got lots of other things going on. And I think from then you're going to throw it back to Emma.
Yeah, so I'll just quickly show you a couple of things. So first is this Never Home Alone project. It's an iNaturalist project. You get this iNaturalist app, you click on it, you sign up, takes 10 seconds. And you just take a picture of the arthropods in your home, and through machine learning and expert IDs, they get ID'd. And we actually use the data. So this project Michael and I read a paper about, it's going to be based on the data that you collect. And so it's super easy. I lead tons of community science projects. I rarely do them. I do this one. It's good, it's easy, it's fun. The other one is that we have a project on sourdough bread. And so if you check out the Great Sourdough Mystery at NC State, or you check out the Rob Dunn lab, you can make a sourdough starter at home and help us figure out what's governing, what makes them super acidic, what makes them rise fast, what makes them different. And we've also got a series of seminars based around that. And so, for example, this Thursday, we got Sandor Katz talking about some new kinds of ferments and what you can do, with the idea that we're always bringing these things together. So science, history, culture, all around a theme. And so those are good places to start. But thank you. It's been an absolute pleasure. And now I will tip back to the Lauren slide, because I know she's appearing. Well, there's Emma. I didn't pay any attention to what I was supposed to do. So Lauren's next. And Lauren's going to tell you how to use the iNaturalist app. Listen up. Lauren also leads a bunch of amazing projects through the museum. And so if you want to get involved in other things, think about a donation. What was the second thing? Just think about a donation again. Remember, if you give money, they give money. It's like getting money back.
Fantastic.
All right, off to Lauren. Thanks, everybody.
You're the best pitch man ever, Rob. Thank you very much.
I know that everyone can see exactly what your face looks like, but I love this photo of you and don't want to miss the opportunity to show it off. So Lauren Marino Perez is the less hairy one in this photo. She is the community engagement manager at the NAT, where she interprets exhibits and scientific research. She enjoys scrolling through iNaturalist on sleepless nights and has helped lead San Diego to City Nature Challenge victory. So City Nature Challenge is a super fun event, which you will hear a little more about in just a moment. Thank you, Lauren.
Thank you, Emma. All right. So, yes, that's me, and this is me, and here we go. So thanks so much for joining us, and if you're still sticking around, I'm really glad that you're here. As Emma mentioned, we're going to take a pause break where we'll answer some questions, and then we'll also do that at the end as well. So you're bound to have questions, especially with a lot of novices getting started. So ask away, no dumb question, please go for it. You can put them in the chat as well. So let's just real quick, what are we doing tonight? What is iNaturalist? Rob mentioned it, Emma mentioned it, Mike mentioned it. how does the Nat use iNat, and then how to contribute. So we're going to learn how to join a project like the one that Rob mentioned, Never Home Alone, and then add an observation to that project. So if you did download the app and you logged in and everything, you'll be able to upload one tonight. And so we're talking about you guys participating in scientific research. And so at the Nat, we call this citizen science or community science. Basically, it's people helping out formal researchers. Like Rob was saying, I think he made an eloquent point of there's so much out there we don't know exists, and scientists can't be everywhere, and we need your help big time. There's still a lot to explore. So that is exactly what we do with community science. It's been with us since our founding as a museum, and we still do it today. So we hope that you'll join us if you haven't yet. And you can actually find more about the projects that we do by going to our website. We're on the events and citizen science, and we'll have this all in a wrap-up email that we share with you all. So that's kind of overview of citizen science and community science. And what is iNaturalist? So iNaturalist is actually a data mapping app, and it's also kind of social media in a way, but it enables anybody with a computer or a smart device, an iPhone, an iPad to document plants and animals, the natural world all around them, and then share it with scientists, share it with other nature lovers, share it with people that don't love nature but are just wanting to see what's in their area or areas they're going to go explore. So there are a lot of ways to use iNaturalist, and we're just going to really tip of the iceberg here tonight. We're just going to show you how to upload an observation, but I highly suggest you get into it more and you go online on the website. There's kind of more capabilities there to play around with it. And I'm happy to answer questions too. I'll have my email, share that out. So feel free to reach out to me. And you will see Levi Passmore here with a giant tarantula on his face. He is an early community scientist that helped the Nat out. And I just love this picture. And I also know Emma can't stand spiders, so I had to throw it in there. There, Emma. But kind of how does the Nat use all this data? Why map all this information? So it gives us a record of what is where, when. And those records change over time. Like Rob mentioned, invasive species, or not even invasive, just new species coming into an area, or maybe things that were in an area no longer being there. Well, if we don't have a record of what was there at a certain time, we don't know if it's changed. So basically, we're just keeping a record of what is living here when. And then we can look back, maybe 100 years from now, people will look back and say, thank God they saved those things because we saw things changed, which they couldn't do otherwise. And I really liked the phrase, what did he use the phrase? Oh, a climate gateway? Is that what he used? That San Diego is a climate gateway. So we're like prime spot for possibly finding new things in North America, especially in our homes, which is super exciting. That got me even more excited about why we shall be participating in this. So the Nat uses all this data. We have museum collections that are basically go back to when we were founded in 1874, and that is mapping all the plants and animals of our region. So we already have all this in databases. And we pull things from iNaturalist, your observations, and we map them. So we combine them with collections data from the museum going back over 100 years. And then we can map these points, and it lets us see where things are and where they have been, and then also look back at how that may have changed over time. So our curators are actively, like Emma mentioned, John Redman in the poll. He is actively pulling in your observations from iNaturalist into his project called the Plant Atlas. And that's mapping every plant in San Diego County. You'll probably see him if you use iNaturalist. He's pretty amazing. I don't think he sleeps, but he is awesome. So let's get going. Let's play with iNaturalist. That's kind of a quick introduction. So you should have already downloaded the app, made an account and logged in, which is just like an email. You're going to be able to log in if you go online to inaturalist.org and log in there and see your stuff, or from any tablet or smart device you do. And then we're going to get started joining projects right now. So in order to join projects, we're going to be looking at this iPhone view right now, and then we're going to switch to Android. So if you have an Android, just hold on a moment, we'll get right to you. It's really, they're both very similar. And if you're used to using your device, they've really set it up so it's kind of intuitive, but it does take a minute to get used to. So on your iPhone, you want to log in and go to the More section, which is the bottom right. 3 little dots for iPhones usually means there's more that you can see, and iPads too. And then you want to choose, that will bring you to projects or guides. You're going to want to choose projects because we're going to be joining projects. Guides are a really neat thing, but they're unfortunately kind of defunct. They are not, iNaturalist is no longer allowing people to make new guides or kind of investing in developing guides, but you can see old ones that were there. And so the idea is it's basically a guide to a certain area that somebody made. Like, you can find this here, you can find that there. A lot of nature centers make them, and I suggest checking them out because they are neat, and you can add those to your account. But we're not going to go into that. We're going to go into projects. So that first little kind of briefcase there, you want to select that. And then it's going to bring up a search icon. It might take a minute. So honestly, don't freak out if it doesn't come up. It's just taking a minute if you don't see that little search icon there. And we want to join Never Home Alone, right? Because Rob just got us all really excited, and we have to add to it and discover new things. And also, I guess basically share like the fact that we're all living in toilets. I don't know, I couldn't get over that. I don't know if anybody else did that. Basically everywhere on Earth is a toilet, but I digress. So you will, once that icon comes up, you can just click in and start typing in Never. So Never Home Alone is the name of the project. iNaturalist is really, really picky. It doesn't like misspellings, and it doesn't like wrong, sometimes they'll have years on things or colons and things, and it wants you to have exactly what is there. So I find it better to do less than more in case you kind of mistype. So I just put in Never to see what would come up. So multiple things come up. The one we want is actually at the very bottom. It has a little spider icon. It's also on the left of this screen here, Never Home Alone, the Wildlife of Homes. Now you can click on these and it will, you can read in a summary about them, and so you could see if you were on the right one or not. And once you do that, you click on it and you hit join, and then if it changes and it shows you leave, that means you have joined it and you are good to go, and it's going to show up in your projects. So iPhone users, you can play around with that. We're going to go to the Android view. Android view is kind of in the opposite, the top left-hand corner. You have 3 bars instead of dots. That's kind of your more menu bar there. You do the same thing. Here you have kind of more options, which is nice. Projects, guides, activity, mission settings. We're going to go to projects. Same thing with the guides. So the guides are being developed more, but they are there, and I suggest checking them out. So we select projects, same thing. You get your little search icon there, and your hand lens, and probably have to take a breath, wait a minute, and then once it's there, hit it, and you can enter in, start searching for Never Home Alone, and we'll click join. But it's going to show you that you've joined something if it says leave here. So that means you're part of it. If you do decide to leave, you are going to lose the observations that you added, just as an aside. So once you join something, you probably don't want to leave it unless you don't mind re-adding your observations. And then once you're on a project, just kind of some fun things. There are different project tools. There's news, and the news is pretty neat. Depending on the project, people are more or less putting things out there, but Never Home Alone usually has some kind of neat articles that they share or blog posts, and this will take you to them. So I recommend checking that out. And then you can also see what's going on, who's uploaded things, how many observations there are. So they're approaching like 15,000. And then how many different species have been added. So over 2,600, how many different people are taking part. And you can click on these things and see more information and scroll through. So it's pretty fun to get lots of info there. So I think now, Emma, do you want to go to questions, you think?
No, we have just one at the moment. It's from Allison, who's looking to find the correct Never Home Alone project. And hopefully you found that, Allison, when Lauren showed the photo and everything. She also asks which projects on iNaturalist should we join? And I think you're getting to that a little later, some of the projects that the Nat scientists use.
Yeah, I have a couple projects I'll recommend, and then we'll also share them out in kind of our wrap-up thank you email as well. But there are a lot of projects out there, so I'm going to just highlight some of the top ones, but that will be at the end. So I'm glad that you asked.
Perfect. So do keep an eye out for that email. Lauren is going to share some great resources with everyone for getting involved in community science projects, and we'll also be sharing a recording of tonight's talk, which you can definitely share with your friends and family, and that email will go out in about a week.
Alrighty, so let's get down to recording observations. Now that we've joined a project, and you will want to join more projects later, like we said, but we're just going to play around with this first one right now. So hopefully during that bathroom break, you may have had time to take a photo of something living in your home, or maybe not living. Actually, Rob had some really great advice when I asked him about it earlier, going to a windowsill and seeing what's in there. I know people have really, really clean houses, so nobody has any insects in their houses, but you know, maybe just tonight by chance something flew in and you can share that with us. Also taking apart your lamp. So when you clean out your lamp, there's going to be a ton of stuff in there. Putting it on a white sheet and then photographing them individually is also a great way for them to see species. So if you do have an observation or some photos you took, play along. If not, just watch. So you are going to go into the app, just your standard view. When you open it up, there's this little camera icon at the bottom for iPhone users, and for Android users, there's a little plus sign in a green circle, and you will click on those to upload an observation. Now, it automatically will take you to your camera in the iPhone, and in the Android, it gives you a couple options. So I personally just don't recommend taking photos with the app itself. You can do that. So this is how you would do that. I like to take photos from my library. And the reason I do that is a lot of times when you are taking photos, you might be out on a trail. Obviously not for Never Home Alone, but I also don't have the best service in my house. And if you lose service, but you only have your photos in the app, then you lose those photos if you aren't able to upload them. If you have them on your phone, you can be in airplane mode and you can still be taking photos and they will be saved. So you don't have to necessarily be anywhere where there's great service. So that's why I just take them on my phone and then I delete them later. So they're not taking up space on my phone. So to do that, you're going to go to your library. So the Android or the iPhone, it's a little library icon on the bottom right there. And then the Android, you're going to tap choose image. And then this will take you to your photo library, and depending, sometimes you can select multiple at one time, sometimes it makes you go back and forth and picking one, but multiple observations, multiple photos per observation is what we really recommend. So you can see I've chosen 3 here of this plant, these white dots at the bottom here that are a little hard to see, but that means there are multiple images, and that's going to help people on the other side identify this for me. So the more images you could get, the better. Talking with Michael Wall even about certain insects, they're hard, they're moving around, but if you can just take multiple photos of them, even if they're a little fuzzy, like ideally clear photos, yes, not always possible. So you want to upload as multiple photos, so that will help people on the other end that are IDing all these things. So there you go, you have your picture up top there. You can tap to the left of it and you can add more from your photo library if you want to keep adding more. And the next thing you go to is enter a species ID. So the nice thing about iNaturalist is you don't have to know what you're looking at all. It will, if you click in that box, the species box, it will actually suggest something for you, and you can agree with that, or you can enter in your own thing. It's not always right. It has really good AI, but it's not always right, or it might focus on the wrong thing. So enter in your own thing or agree with it, but always enter something. If you don't enter something, people that are going through, earlier you might have heard Rob and Michael talking about how, you know, they have somebody doing just the beetles. Well, he's just searching for beetles. He's not looking at anything else. So he's not going to bring up something that says unknown. So if you want to get your photos in front of people that are the experts, you want to add something there. So just saying beetle totally works. Just saying insect is perfect too. Plant, there are people on the other side just waiting to jump on an ID. So it's awesome. But if you don't ID it, then months, years can go by and nobody ever looks at it just because it's not coming up in their searches. So you click in there, always suggest something, and a lovely little rattlesnake. And then also, if you start identifying things and you realize that you like identifying while you're observing, you can go in and identify for other people too. So it's totally peer-to-peer. As long as two people agree on an ID, then it becomes research grade and researchers can pull that into their projects. But people can also overwrite things and say, I disagree. And there's some really interesting conversations that you'll see between people disagreeing on identification. So I oftentimes will just say lizard, plant, because I don't know, or I'm not sure. You can add notes. So this is a kind of, might be a fun place to add notes. Actually, some of those questions that came up might not be explicit from your picture, but you could put in here, found in shoe, or in my bathroom, or things like that, that also might give people clues or that want to know more about where you found it. It also kind of acts as your own little personal journal because you can access these later. And so if you want to write like north side of the hill or cloudy day, just notes for yourself too, to kind of remember. This is a great place for them. And then if there are multiple organisms in the photo, you want to do an observation for each organism. So this was actually, I took this photo on a plant at a nursery, and there's a caterpillar, and then the fly just happened to be there as I took the photo. So I would want to do 2 different observations, one for the caterpillar and one for the fly. The reason I'm not doing the plant is just because it's a planted plant at a nursery. It's not a wild plant. If I did want to do one for the plant, if I wanted to try and get it ID'd because I didn't know, I would just mark it as captive cultivated, and I'll show you how to do that in a minute. So this next one is important to people, location. So when you take a photo on a smartphone, a tablet, a lot of digital cameras too, it's automatically tagging where you are, your latitude and longitude, and that's just in the metadata in the photo, and it will upload. So you don't have to put your location in here. When you upload that photo from your phone, it's going to automatically upload. You can see here on the left-hand side, this is some kelp, and it was taken at Coronado Beach. And the lat and longitude are there, and the geoprivacy right below says open. So anybody that's on iNaturalist can see exactly where I took that. Well, as you might imagine, if you're taking photos in your house, you might not want people to know exactly where you are. So here, if you go to that geoprivacy, you can select it and then hit obscured. We recommend obscured as opposed to private, because obscured means the people running the project can still see it and you can still see it, but nobody else can see it unless they're a trusted user that you've friended on iNaturalist. If you go to private, then nobody can see it, and it isn't as useful for a research project. They can reach out to you and ask, hey, where did you see this? But if it's not something truly remarkable, not to say not everything is remarkable, but if it's not something that they really, really need, they may not have that grad student that has the time to reach out to you and say, hey, where did you find this? So obscured is a way to just make it accessible to the people running the project, yourself, while also keeping your exact location safe. And I also really recommend this if you're going out with people under the age of 18, obscure observations. It's just an easy way to be safe, and it still works for everybody, still research grade. So these are what the different geoprivacy settings look like. You'll see actual, if people can see exactly where it is, it's like a real map, like dot on a map right there, pinpoints in. If it's obscured, it's going to be round and big, and it actually throws it off by miles and miles. So it kind of depends on where you are in relation to the like poles and how much it obscures it, but it obscures it. So. And so as I mentioned before, if you find something that you want ID'd that isn't wild, something, you know, in your neighbor's yard that maybe you want to eat or something, you can select captive or cultivate. So it'll still get an ID, but people know that there isn't a wild plant growing somewhere that it doesn't belong. So I don't know, I say like passion flower or something like that, which could just pop up somewhere, be blowing in or something. But if it's just in your neighbor's yard and they planted it and it didn't just pop up on its own, then it's cultivated. If you come across like a dog or a bird or something that is obviously somebody's pet, that's captive. So people don't really need this for research purposes. So we're not asking people to upload captive or cultivated things, but you still can. You just ask that you for sure click this box so researchers aren't confused as to why there are wild parrots flying around San Diego, which there are, and they used to not be here. So this is a great little box to check so we're not being confusing to people. And then the really important part, if you want your observation to be added to projects like Never Home Alone, you will select at the very bottom projects. And then that will bring up all your projects. So right now, if you just added Home Alone, that's the only project that's going to pop up in your project list. You're going to click on that and it will turn green. That's how you know you've added your observation to that project. And then when you're all ready, we recommend doing this probably when we're back in a service area. You can just tap share on an iPhone, on an Android. There's a little check that you tap, and then it will start uploading all these guys for you. So a couple of best practices, as I mentioned, you want to take multiple pictures for each individual organism that you're recording in an observation. So various angles, same with plants. If you can get up close, if you can get the full animal, the full plant, also get up close. Different angles, especially with birds, are hard, but silhouettes can help and angles can help. If it's something that you can get your finger in there, if it's small enough for scale, that's also really great, as long as you know that you're safe and you're not touching a poisonous plant or getting too close to a wild animal. Those are all great things to do. You want to take photos of one individual organism per observation. So make observations for each different one. If there are multiple in an image, like in the background of this, we've got a poppy, we've got a bee, you would do one for each of those. Same photo, but just call it out in your notes. This one's for the bee, other one's for the puppy. Or this one's for the insect, other one's for the ant. Best practices also, we want those wild plants and animals ideally. Mark anything as captive and cultivated if it's not wild. And then projects to join. So the NAT has a number of great projects. We just talked about Never Home Alone, not the NAT's project, but Mike Wall is helping out with it. And it's really an awesome project, and we need to get the numbers up in San Diego for sure. We're not pathetic, but we're not great, and we could be great, San Diego. I know we could be. Bumblebees of San Diego County is a really fun one. This one's documenting different bumblebees around San Diego, and that is one that actually Michael Wall administers. That's run by the NAT, the San Diego County Plant Atlas and the Imperial County Plant Atlas. Those two are both administered by our curator of botany, John Redman. And then we have the Rascals Project, which is actually the NAT in partnership with the Natural History Museum of Los Angeles County. And curator Brad Hollingsworth, he pulls those observations into his herp atlas. So as I mentioned before, we have the museum collections and then the iNaturalist collections together to kind of give us that really complete picture. So there are those. There's a lot more out there. You can go to our sdnat.org/citizen science page, and you'll be able to find even more projects there that you can join. And then oftentimes those projects will lead to more projects. So And then that's my last slide, unless we have any questions.
We do have a few questions, and I actually want to start with one of my own. Thank you so much, Lauren, for going through this. You've gotten me really excited about these 5 projects here. What happens if I observe something that isn't a good fit for one of these projects? For example, a squirrel out in the wild. Where does that observation go? Is it legitimate to make?
Yeah, that is a great question. So actually, you can still make the observation. People are still going to ID it. It still is going into this resource of data, and then people running different projects might find it. So if you tag something as a squirrel, people that are running projects about squirrels may be searching on the back end and might find your squirrel and pull it into their project. But so it's just great to have it up there. You don't have to be adding to any projects. But there are certain projects like Never Home Alone that you really do want to add to, because otherwise that's probably not going to be seen by the project because there are so many observations.
Great, thank you. On the subject of Never Home Alone, we actually got a question through the chat from Dr. McHager, who was the president of the NAT for many, many years. So it's great to see his name pop up. He was curious, for the Never Home Alone project, are we only doing observations indoors? Does the driveway count, or the front yard?
Yeah, that is a great question. So my understanding with talking with Dr. Dunn is inside the walls of your house, inside the house. Yeah, I know. But anything in your yard is great also to just upload those, and those will fall into all sorts of different projects. You don't even have to have added them. There's a project that's just San Diego County biodiversity that no matter what you upload in San Diego County goes into it already. So just observing in San Diego County, you're already adding into a project, and you don't have to join that one. It just happens automatically. And then, if you find a lizard, it may not go into Never Home Alone, but Brad might want it for his Herp Atlas and the Rascals project. So I always say just add them.
Great, thank you.
Mm-hmm.
Allison is wondering, how do you delete an observation? She doesn't see a trash icon.
Yes, there isn't one, but if you go to, let me bring back images. You should be able to go from kind of your landing page. If you select an actual observation itself, there is an edit button. So this top edit button, and that is where you can delete it, is through there. That'll bring you to it.
And then Peter actually has some really useful information that he wanted to share with the group, which is that any recent evidence of an organism counts as an observation on INAT. So scat, tracks, nests, shells, beaver chews, molts, etc. are all fine. I've definitely personally enjoyed doing scat observations. I always am convinced it's a mountain lion. It never is. It's about the only mammal observations I've ever made.
It's a reoccurring theme that the world is a toilet, I guess. But I was also going to say, if you do end up taking evidence of animal life, having something in there for scale is really helpful. So with scat, definitely having, if you have like a quarter in your pocket, popping that out next to it if you don't want to put your hand next to it, footprints, things like that. Something for scale is super, super helpful. And as I mentioned before, things don't have to be alive. Like taking all the dead moths out of your lamp totally counts. So, and really any evidence of plant or animal life is totally great.
Great, thank you.
Yeah, thanks for pointing that out, Peter.
Thanks, Peter. Michelle is wondering if iNaturalist is just a US app or if it is worldwide.
So it is worldwide. It has different names in different places. And if you are in a different country, when you download the app, it'll prompt you to use that. So in Mexico, it's iNaturalista, and there are different affiliate names throughout the world. But also if it's, say, you're an American user traveling in another country, you will just stay with your network iNaturalist and upload it on there. But if you're a user in Mexico, you'd use iNaturalista, and that will upload to iNat as well, and it works both ways. So they all pull from each other. They just are different platforms in different countries, if that makes sense. And then they're automatically translated as well.
Great, thank you. And we've mentioned the City Nature Challenge. That is a worldwide competition. It's really cool to see the countries we're up against. And actually, I think La Paz, Bolivia was maybe one of the winners last year. Am I remembering that right? South Africa has done really well.
Yes, Cape Town does really well. I believe La Paz, yeah. It feels like, I'm like, was that last year or 10 years ago? Because it was in April. Seems like another world away, a lifetime away. But yeah, it is. It's really fun. You can get to participate and compete against all sorts of other cities, including LA. So.
Need LA. Next year's our year. Dan is wondering about the back end. He asks, is there a way to start a project and have observations from others automatically add to that project if it fits the parameters of the project?
Yeah, so when you do go to the projects page, to start a project, you cannot do it on the app. You need to be online on inaturalist.org. So there's a lot more functionality there. And you would start something called an umbrella project. So that is, you're running kind of an overarching umbrella project, and it's pulling in from different projects. So they can be location-based or species-based or date-based. There's all sorts of different options. And there's also all sorts of tutorials on there as well, and videos, and really some great help forums. People are super helpful on there if you just ask questions. In the app itself too, I meant to point out to everybody, or you know what, let me click, it's towards the end. In your settings, so the top right here is your settings. If you go into there, there is a tutorial on getting started with iNaturalist that you might want to check out as well.
Cool, thank you, Lauren. It's getting late, so I just want to wrap up with one final question, which is, can you tell us just one or two instances in which iNaturalist observations have been really impactful on research?
Well, I have a couple, I guess, examples just from the NAT itself, one of them being a newly documented behavior as a part of the Herp Atlas and the Rascals project, where a coyote was shown predating on, so eating a rattlesnake, a red diamond rattlesnake, which hadn't been documented before, but somebody took a photo of it, got a really cool photo of it. You could totally tell it's a red diamond. It's got those white stripes on its tail, and there's a coyote with it. And then also with the Herp Atlas, now that I think about it, an example of new behavior for, it's a king snake, predating on bird eggs that it hadn't been recorded as doing before. And so that was a publication came out of that, which is really exciting, because the citizen scientist, the community scientist that observed it, was actually a part of the research paper. And that will be the same actually with Never Home Alone. Huge contributors, people that are doing like large number of contributions to that, will be a part of the research project and the publication as well. So that's super exciting, I think, to get to really see how you're adding to this collective body of knowledge that we're all learning from, and so much to learn from.
That is so stinking cool.